Hello friends. So today we will start with another system of our body which is related to brain. So up till now we have studied about the nervous system, and then we have discussed about the electrical activity of the heart and ECG signal which is related to our heart, and in this we will see about EEG signal, that is electroencephalogram, which is related to our brain. And then like we have seen how we are recording ECG signal, in this we will see how we will record the EEG signal. So let's begin first with the introduction to brain. What are the main parts of brain? So the brain is one of the largest organ in the body, and it coordinates most body activities. The human brain, we can say it is the powerhouse and control center for various physiological and psychological functions like consciousness, mobility, emotion, personality, etc. So all these systems or functions of our body are controlled by the brain. The brain contains approximately 100 billion neurons that work together to carry out complex functions. Now let's discuss what are the in brief we will discuss what are the main parts of the brain. So first is, we can say that brain is the main switching unit of the central nervous system, and it is a place from which the impulse originate and to which impulse from the different parts of the body reaches. This also we have discussed in the part of central nervous system, because brain is the main part in central nervous system. So what are the main parts of the brain? We have cerebrum. You can see in the figure also it shows typical structure of the brain. Then we have cerebellum. Further, we have brain stem, which includes midbrain, pons, and medulla oblongata. So we have, these are the main, or we can say, journal parts comprising the brain. Now we'll discuss one by one all these parts. So first is cerebrum. It is the largest and the most prominent part of our brain. It is responsible for all the voluntary means conscious activities of the body. That also we have seen. We have two kind of responses. One is voluntary, other is involuntary. The cerebrum is responsible for all the voluntary activities of the body. And it is further divided into two hemispheres. We have left and right cerebral hemispheres. Each hemisphere of the cerebrum is divided into four regions, which is known as lobes, that are responsible for different brain functions. So now let's discuss about the four lobes of cerebrum. So this figure shows the four lobes, and they are intentionally shown by different colors. So we have frontal lobe, we have parietal lobe, we have temporal lobe, and occipital lobe. So these are the four lobes of the cerebrum. Now we will discuss one by one each of them. So first is the frontal lobe. It is located, as the name says, it is located at the front of the brain. It is the area of the brain responsible for higher cognitive functions, including reasoning, motor skills, judgment, and expressive language. Then we have the parietal lobe. It is located in the middle section of the brain. It is associated with processing sensory information like touch, smell, and taste. So mainly it is related to sensory receptors. Then we have the temporal lobe. It is located on the bottom section of the brain. It is important for interpreting sounds and language we hear. This again we will refer when we will discuss about the auditory system. So in that also we will take that temporal lobe. It is used, or it is mainly important for interpreting sounds and the language we hear. Then we have the occipital lobe. It is located at the back portion of the brain. You can see in the figure. So it is associated with interpreting visual stimuli and information. So this lobe we will consider when we will discuss about the visual system of our body. So these were the four different lobes of the cerebrum. Now the next main part of the brain is cerebellum. You can see, so it is the second largest part of the brain, and again it is located at the back of the skull. You can see in the diagram it is showing cerebellum, which is located at the back of the skull, and it is useful for coordinating the muscle movements. It controls the balance, posture and coordination of the body. This again we will refer when we will discuss about the muscular system of our body. So it receives sensory impulses from muscles, tendons, joints, eyes and ears. Next part is the brain stem. As we mentioned earlier, it consists of midbrain, pons and medulla oblongata. So it connects the brain to the spinal cord. That is another composition of central nervous system, because we know in central nervous system we have brain and spinal cord. So this brain stem basically connects the brain to the spinal cord. It controls the vital body processes, and the brain stem, in that we have midbrain as we mentioned, then pons and medulla oblongata. So midbrain, it controls many important functions, such as we can say the visual and auditory system as well as the eye movement. Then pons, they control sleep as well as the rate and pattern of breathing. We have next is medulla oblongata. It controls automatic actions such as breathing, heart rate, swallowing and blood circulation. So all these involuntary, we can say involuntary actions are controlled by medulla oblongata. Now, let's start with the electrical activity of the brain. Now, the brain consists of billions of cells, half of which are neurons and half of which help and facilitate the activity of neurons. So, if we have millions or billions of cells, in that half of the neurons help and facilitate the functioning of neurons and other half are neurons. Now, let's discuss the working of that. Neurons are electrically charged by membrane transport proteins that pump ions across their membrane. These neurons are densely interconnected via synapses. This also we have studied in the lecture of nervous system and neuron. These neurons then act as, we can say, gateways of inhibitory or excitatory activity. So this is showing the synapse, you can see. Now let's discuss about the sources of brain potential. Any synaptic activity generates a subtle electrical impulse, which is referred to as a postsynaptic potential. The burst of a single neuron is difficult to reliably detect without direct contact with it. So, but because we have thousands of neurons which are firing in synchronization, so they generate an electrical potential which is strong enough, and it is spread through tissue, bone and skull. And when this wave of ions reaches electrodes on the scalp, because how we are capturing the activity of the brain, we have to keep different electrodes on the scalp surface. So when the wave of ions reaches these electrodes on the scalp, they can push or pull electrons on the metal of the electrodes. And this push or pull difference measured as voltage across time, and that can be measured on the head surface. Let's discuss this in more little bit detail. So you can see this is cerebral cortex. It is the outer layer of nerve tissue. and it is located in the cerebrum of the brain. Now, in cerebral cortex, there are specialized neurons and they act as pyramidal cells. These are specialized neurons and we call them as pyramidal cells. And some of them are relatively large and they are arranged parallel to each other and perpendicular to the surface of the brain. Now, what happens when action potential propagates along the neurons? You can see we have studied this thing when this action potential begins. So when action potential propagates along the neurons, then local currents are produced outside of the cell which facilitate propagation of the signal along the neuron, and however these currents are too small to be detected by EEG. And also we have seen the axons can be arranged randomly. So many of the currents cancelled out, but there are other sources of currents in neurons. Now, as the synapse between two neurons, we know that neurotransmitter, you can see these neurotransmitters, move across the synaptic cleft and binds to the postsynaptic membrane. So, and that because of that, it causes ion channels to open in a membrane and positively charged ions flow into the cell with the extracellular space around the neurons becoming negatively charged, and it is referred to as sink. And then in distant part of the neuron, the ions eventually leave the cell, and this outward flow of, we can say, positive ions leaves the extracellular space positively charged, and this is referred as source. So this combination of two related processes, they forms a dipole. So it forms a dipole between different parts of the neuron. However, the dipole from a single neuron is very small and there is a thick skull as well as the outer layer of soft tissue. So they have to pass through this individual, we can say, different regions of brain. So individually if we talk, they are undetectable. But as we know, there are so many pyramidal cells in this layer of the brain. All are aligned parallel to each other, and often many of these cells are stimulated at the same time. Therefore, this is detectable outside the head by the help of electrodes. So this was how the potential generates in the brain, which can be measured by the electrode placed on the skull of the brain. Now, next is electroencephalogram. That is a signal which is related to the brain, or we can say the functioning of brain. So whenever we want, like we have to test whether our mind or brain is working properly or not, we have to go for EEG. So it is a method to record the electrical activity generated by the brain via electrodes placed on the scalp surface. So these are the electrodes, you can see they are placed on the scalp surface. It measures electrical activity generated by the synchronized activity of thousands of neurons, mainly volts. So these first recordings were made by Heinz Berger in 1929, and during the EEG test, this cup or disc type electrodes are placed on the scalp, and these electrodes pick up the brain's electrical signals and send the same to be recorded on the electroencephalogram. And these are generally the voltage fluctuations measured at the electrodes. They are very small, so the recorded data is digitized and sent to an amplifier. And this amplified data then can be displayed as a sequence of voltage values. So this was all in this. Now in next lecture we will discuss more about EEG signals, how we can record that signal, what is the 1020 electrode system to measure EEG signal, and further we will analyze EEG signal for a normal person as well as for some kind of abnormalities. So for now, thank you so much.